A very good evening, all of you. Today we are going to discuss about the phylum Porifera. The phylum Porifera, generally we call it as a sponges. Phylum Porifera, the name was given by Robert Grant. Robert Grant in 1825. Now, let us see the characteristic features of poriferans today, and in the next class or upcoming video we will see the classification and examples of poriferans or the pore bearers or sponges. Actually these poriferans are called pore bearers, right? Because all the body is composed of pores only. We will see that discussion also in the upcoming topic. We will see the characteristic features first. The habitat. Mostly these poriferans survive in marine condition. In marine condition, in salt water only we can see. Freshwater, very rarely we can see the freshwater sponges. Like spongilla is the example. Then these sponges we fail to found in cold water. We will see sponges only in the warm water. They like staying in the warm water, not in the cold water. Coming to the body form. Body form, as I told, that it is a porous, that's why it is called as pore bearers. And the porous, two type of pores we can see, that is inhalant pore, exhalant pore. Inhalant pore is ostium, where the water can come in. Exhalant pore is osculum, where the water goes out. If you see, this is what ostium, the water comes in this way, so water will come this way, and this is osculum, where the water goes out. So this is how water canal system is well established in sponges. So if you see this pore, through this aperture the water will come in. This aperture is called, I labeled here, it is ostium, and water comes into the spongocele. Since it is coelomate, so coelom is present, so spongocele we call, the coelom present in the sponges is called spongocele, and the water enters into the spongocele through ostium and goes out through osculum. Ostium, osculum, right? Then symmetry. Actually the adult is asymmetry because it's irregular growth, and the branches also will be in the sponges. If you can see in the diagram of NCRT, you will understand that. Then the larva shows radial symmetry in a few. Then the germ layers. In the embryonic layers we can see them with only two layers. That's why it is called as diploblastic, the outermost ectoderm and the innermost endoderm. We'll see what is that ectoderm and endoderm also here. And the levels of organization of the body of these sponges is cellular level. See, the sponges fail to form the tissue to conduct different type of functions. Sponges, they conduct their individual functions by individual cells itself. How the functions will be conducted, I listed here. We'll discuss that also. So different type of functions, like for example in the human being, if you see, the excretion, osmoregulation is conducted by the kidneys, organs. Excretion is conducted by the kidneys and the skin also. So here different type of functions are carried by different type of tissue, organ and organ system in the human. But in the sponges, if you see, there is no tissue, there is no organ, and organ system is forming to conduct different type of functions. So functions are carried by the individual cells itself. That's why the levels of organization is cellular level of organization. And the body wall, as I told that here ectoderm and endoderm here, the body wall consists of three layers, actually two true layers and one is false layer. We'll see the outermost, this red color layer is called pinacoderm or pinacoderm. It's a dermal layer we call. Dermal means outermost, the covering layer is a dermal layer. It is a red color is pinacoderm. The cells in the pinacoderm are called pinacocytes. And apart from pinacocytes, the ectoderm or else this pinacoderm will have the oval shaped, I labeled here the oval shaped, I just here myocytes, I didn't label it. This black color cells are called myocytes. So pinacoderm will have flattened pinacocytes and ovular myocytes waited. Okay, these myocytes actually, okay, myocytes especially present in this area. Let us discuss that also. And the choanoderm is, if you see, choanoderm, the innermost blue color layer is called the choanoderm. Here, I am sorry, here these are not called myocytes, these are called porocytes. Porocytes. So here, the flattened pinacocytes and oval porocytes are present here. Porocytes which help in the opening and closing of ostium. Then choanoderm, the blue color layer, if you see, this is the choanoderm. The cells present in the choanoderm are called collar cells, or else called choanocytes we call. Choanocytes helps in the trapping of different type of food material or else the prey. Then the false layer, I told this in between ecto and endoderm, in between pinacoderm and choanoderm, there is a green color cells are present. They are actually, this is called as mesenchyme cell or else mesohyl layer. I labeled here mesohyl layer or else mesenchyme layer we call. This layer consists of number of cells which will carry different type of functions we can see here. So this mesenchyme actually, especially it is designed with amoebocytes. These amoebocytes again modified into, they are differentiated into different type of cells. So commonly all the cells are called initially amoebocytes. Later on amoebocytes will be differentiated into these many type of cells. Let us see each cell functioning. If you see first one, archaeocytes, in this mesohyl layer, what are the cells are present is first one archaeocytes. They help in totipotent, they are very sensitive to touch, they are sensitive in touching. Then second cell, trophocytes. Trophocytes actually they provide food material to the different type of cells, that's why they are called as nurse cells. Third one, thesocytes. Thesocytes, they store the food granules. And fourth one, gland cells, they secrete different type of slimy substances which will help for different functions in the sponges. And colonocytes, they actually secrete spongin fibers. Spongin fibers is one is the type of the skeletal model in these sponges. Then scleroblast secretes spicules. The spicules may be the siliceous. If it is a calcareous sponges, we call it's called as a calcaroblast. If it is a siliceous, we call it as a spicule. If it is a calcareous sponges, we call it as a calcaroblast. Then the next cell, myocytes. Here I told, actually I mislabeled here and I am telling here myocytes. Myocytes are the cells which are present around the osculum. Osculum is the exit of or exhalent pore, it is exhalent pore where the water goes out. So osculum is surrounded by myocytes. These myocytes will help in the closing and opening of this osculum. Okay. Then germ cells. In the mesohyl layer itself, sex cells are present which will produce the gametes like male and female gametes, which will help in the sexual reproduction, that we will discuss in the latter topic. Then coming to chromocytes. Chromocyte helps in the pigmentation or coloration of sponges is one reason, and the second functioning is they help in excreting different type of substances also. Then phagocytes. Phagocytes, they receive the food material from the choanoderm or choanocytes and they engulf. These phagocytes are present in the mesohyl layer and they receive the food material from the choanocytes and they engulf, and they even engulf excreta and or waste substances which are present in the inner surface and they'll excrete. That's how the phagocytes. And coming to the skeletal system. Right. The skeletal system is composed of spongin fibers, otherwise the siliceous or calcareous spicules. So the silica containing spicules or calcium containing spicules. Then the type of digestion will be the intracellular digestion. How the digestion will take place in amoeba inside the food vacuole, such type of the digestion will take place here. Then circulation. The water vascular system is well established in the sponges. What is this water vascular system? Let us see here. Then here the water vascular system is water comes in through this ostium into the spongiocele. When the water comes in, it will bring the oxygen and food material also. Then oxygen will be utilized by this innermost layer, and food material also will be trapped by these chromatocytes or phagocytes, whatever, and the digestion will take place there. And when the water is going out, it will take out the extra gases or unnecessary gases and excretory material also. So this type of the circulation is called water canal system. So here the blood is not established, that's why the water canal system is present here. And then coming to the next one, the respiration and excretion. Respiration and excretion will be carried by the process diffusion according to the concentration present of different type of materials inside, and this diffusion takes place through plasma membrane or cell membrane of every cell, because the organization itself is cellular organization, so it's not difficult to conduct a diffusion process for the respiration and excretion. Coming to the next point, the reproduction. Reproduction will take place through sexual reproduction and asexual reproduction also. If it is asexual reproduction, it will take place through budding and gemmules. These are the gemmules. Here the gemmules, during unfavorable condition, what will happen is the sponges will form two layers, outermost layer, outer membrane and inner membrane. Inside this inner membrane, there is the mug-like structure, the jug-like structure is present inside, and all the living cells of these sponges will be present inside and strictly protected by outer and inner membrane and even to the spicules. And these cells, the innermost living cells are protected by these two membranes are called, actually these are archaeocytes. During favorable condition, these archaeocytes will be emitted out through micropyle and will be germinated into the new sponges. That's how the asexual reproduction takes place. And then coming to the sexual reproduction. Sexual reproduction, male and actually they are hermaphrodites. The sponges are hermaphrodites, means the male and female gametes will be present in one organism. So they are hermaphrodites, and the development will be indirect development. Why? Because in their life cycle there are larvae. There is a larva. For example, if you see the cycon organism will consist of amphiblastula. Leucosolenia organism consists of the parenchyma. And here the development, the holoblastic division takes place, or holoblastic cleavage takes place in the zygote. Means the whole zygote will be divided in equal amount. That's how that type of division is called holoblastic cleavage vehicle. And in the upcoming class we discuss about the classification and the examples of sponges. Thank you.